Not everyone believed that protein was the genetic material. Brings us back to the puzzle. And a second piece. Frederick Griffith.
I read about Frederick Griffith. He was really important, but I can't remember why.
Frederick Griffith was a bacteriologist working in England in 1928. He was trying to produce a vaccine against pneumonia. He experimented with 2 different strains of the bacterium Streptococcus pneumoniae. Let's take a look at his experiment. Frederick Griffith used 2 strains of the bacterium Streptococcus pneumoniae, an encapsulated strain which causes pneumonia, and a non-encapsulated strain which does not cause pneumonia. The capsule protects the bacterium from the immune system of the invaded organism. When he injected one group of mice with encapsulated bacteria, they contracted pneumonia and died. Griffith took blood samples from the dead mice and found live encapsulated bacteria in the samples. He injected a second group with non-encapsulated bacteria, which produced no infection, and the mice lived. Their blood samples revealed no live bacteria. Then a third group was injected with encapsulated bacteria that had been heat-killed. In this case, the mice also survived. The application of heat had apparently weakened the virulence, or the pneumonia-causing capacity of the encapsulated bacteria. However, when a fourth group was injected with a mixture of heat-killed encapsulated bacteria and live non-encapsulated bacteria, the mice died. Individually, the heat-killed encapsulated bacteria and the live non-encapsulated bacteria would not infect the mice, but in combination, they killed the animals. Blood samples from these mice revealed live encapsulated bacteria in the mice. What conclusions do you think Griffith made from these results?
Well, there may have been something still present in the non-encapsulated bacteria which caused the infection, even though the encapsulated bacteria were heat-killed.
And the bacteria was killed, because in this one the mouse is still alive. But the mouse that was infected with the non-heat-killed encapsulated bacteria is quite dead.
Yes, the non-encapsulated bacteria was alive, so therefore it may have been able to acquire the ability to cause the infection. Even though the heat-killed bacteria were dead, genetic material that can cause pneumonia may have still been functional, and it therefore was passed on to the live bacteria that infected the mice.
Great deduction, Benez. The principle of living organisms incorporating extracellular DNA in their genome is called transformation.